Okay, so I want to give everyone the welcome to today's colloquium. Today is our last colloquium, Alexa488, from the program that we have been developing with the Montreal Museum of Fine Arts during this last year, but we have been working on it for the last 2 years and something. So came the pandemic and we have to change everything from a in presentation, right? Something that we were going to do in the museum, to do it all online. And it's been a fantastic experience. We have learned a lot. We are very happy that this has been very successful and very thankful to to all our partners and collaborators also. So a part of us, right, Convergence, the Convergence Initiative and the Montreal Museum of Fine Arts working in this, I have to also acknowledge and thanks the support from the brain program, the brain repair and integrative neuroscience program of the McGill University Health Center Research Institute, who has been behind us, helping us in all the process. Also, Healthy Brain for Healthy Lives program from McGill University, who has supported us with funds. Concordia University, who made possible for us to use the Zoom platform for connect everyone from everywhere to these colloquiums, to the Canadian Association for Neuroscience, and to Canada's first Research Excellence Fund that also has been supporting us. So we are very happy to be able to complete this series of talks that cover science, aspects of science, vision science, with fine arts, with a special curated collection from the Montreal Museum of Fine Arts to talk about this. And this is our last colloquium today, and we will have our last workshop next week uh, in the hands of Jihan Moslin, an educator, an art educator. And we are hoping really that you have enjoyed this. We will soon launch all the things that we have done here in our platform in Convergence for you to go and enjoy it again and see it again and participate in the workshops again if you want, or if you have missed any of these past events. So that's from me right now. I'm really happy. I'm very thankful for all the participation of the people during these last 5 months. And I'm going to give the ball to my friend Stefan Legari now. So, Stefan, all yours.
Thank you, Christian. My name is Stephen Legary. I'm Program Officer for Art Therapy at the Montreal Museum of Fine Arts in the Division of Education and Well-Being. And under our division, we have the great pleasure and privilege of having a number of dedicated volunteer guides who commit their time to connecting people to the fine arts through their own passions, curiosities, and interests. And we've been really lucky in this series that they've been so interested and invested in exploring what we can provide online. We don't have access to the entire collection. There are often things called copyrights involved. So we've gathered together the works that we can show you and really explore them in detail. And I'm grateful to each and every one of the guides that has participated in this series. And what a moment to arrive at the end of this series too. Here we are in mid-August. What a perfect time to stop and express a little gratitude to Convergence. You're seeing Christian on screen right now, but also a debt of gratitude to the Convergence team who are not on screen right now, who've invested a lot of their time in constructing this series and do a great deal of public education and bringing together the sciences and the arts, which once upon a time weren't separate. And so this is a gesture of reuniting the humanities and the sciences once again. So I'm really privileged to be here with you. I'm going to put in the chat, oh, look at that. There's the workshops. You can see the workshops. And I'm going to put my own 2 cents and invite you to explore the tours. So in the chat, you will see a connection to the workshop, the final workshop that's coming up next week. And you will see a connection, a link to the tours as well. Tours are also the labor of love of our volunteer guides. And I encourage you to just slow down and have a look at that tour because they really are a beautiful way of exploring in rich detail the artworks that we're presenting to you.
Stefan,
yes.
I would just like to give, before I forget, just a big shout out to Cecilia Kramer for all the amazing work that she has done, not just in these 5 months, but during all this time working with us as a communications officer of Convergence. She has been just absolutely amazing. Cecilia, thank you so much. You are just one wonderful person to work with, and this would not have been possible without your work. So thank you.
Thank you to all of you. Like, this is my pleasure, and I enjoy every single colloquium and workshop. So I encourage everyone to go to the website and check the previous colloquiums if you haven't done it. They're going to be uploaded soon. So thank you so much for giving me the opportunity to Christian and Stefan.
I will certainly extend my thanks to Cecilia as well. If this is going well, it's because of the people behind the scenes. Without further ado, let me introduce our volunteer guide today, who's going to bring you in the fantastic world of the fine arts. Madeleine Colasson, a volunteer guide at the Montreal Museum of Fine Arts since 2018. For her, one of the most enjoyable things about guiding is the variety of conversations that art prompts and the bridge it provides for bringing together different points of view. As a professional marketer and fundraiser, she had benefited from the enrichment that arises when seemingly unrelated paths converge. After graduating from McGill University with an MBA, Madeleine worked in marketing for major consumer packaging goods companies in Montreal and London, England. A job managing sponsorships for the Heineken beer brand became an unlikely bridge into evaluating sponsorships at the Canadian Space Agency. This, in turn, led to a role in fundraising and sponsorship at the CHU Saint-Justine University Hospital Center. She is delighted to be part of the Parallel Worlds initiative, where current knowledge about color and visual perception is explored through discussion between the scientific and art communities, as well as the broader public. I would like to turn it over to Madeleine, and I hope that you all enjoy her presentation.
Here I am. Thanks very much for the introduction, Stephen, and good evening, everybody. And I hope you're sitting somewhere cool. I'm trying to delay the moment of truth, which is my screen share, which I hope is going to work seamlessly. And there, I hope you can all see that well. Yes, wonderful. So yes, I'm delighted to be here with you this evening. And as Stephen mentioned, I'll be focusing on the art portion. More specifically, I'll really be looking at 3 artworks and talking a little bit about these works from the perspective of technique, but also how in their time the artists and the art they produced was pushing new boundaries and pushing innovation and new ideas. And this is really also the link with Alexa488 in fluorescence microscopy, because as Melina will explore in great detail, there was a lot of innovation and newness there. So that's certainly a bridge between our 2 parallel worlds. And I hope that during the course of the 2 presentations, you'll find some more that we can discuss later on. So without further ado, I'm going to get going and hopefully put you in the mood for some art by showing you this picture I took on Avenue du Musée in Montreal, for those of you that aren't from the city, showing various of the pavilions of the Montreal Museum of Fine Arts, which has 5 pavilions. The main entrance, is down at the bottom of the avenue. And the works that I'll be discussing are from the Quebec and Canadian collection of the Montreal Museum of Fine Arts, which is in this building over here. And I'd like to do also a little shout out to let you know that there are actually some wonderful in-person tours, small tours outside of the 5 pavilions of the museum, the architecture of the pavilions, as well as our sculpture garden. So if you're looking for something to do, they go into September, a wonderful way to spend an hour or so of your time with our guides visiting the museum outside. So the first work that I'm going to talk about is this work, which is a stone cut engraving by an artist called Parr, who's from Nunavut, or who was from Nunavut, which so in northern, the Arctic region of Canada. And it was done in 1961. And what I'd like to talk about first is really how even with a very simple black line drawing, you might say childlike, that might have been your first impression. What are some of the elements that we might use to construct the narrative? And of course, different people may construct different narratives, but we'll probably look at some similar elements. So for example, you may have noticed we have 5 human figures and 5 animals, and we'll look at size. So you'll see there's 2 larger human figures and 3 smaller ones. So you might infer a family. And indeed, Parr did call this stone cut engraving les miens, or my ones, indicating a unit. And you'll see that they've all been placed together as a cohesive unit, including the animals. So we have this sense of cohesiveness. We also have quite a lot of expressiveness, even in these horizontal and vertical hand gestures. Very tiny hands, you may have noticed, almost like flippers on animals. I don't know if there was some desire to, again, reinforce the closeness between the animals and the humans. Here in the clothing, we have diagonal lines and vertical lines, which give us a sense of movement too. We have a little curve here. So under these tent-like garments, we get a sense of the body shape. And you'll notice that the animals all have these horizontal lines, and often horizontal lines give a more stationary impression. But clearly they better get a move on, because it does look like Junior is about to practice his or her hunting skills. And this notion of hunting is a way to lead in to talk a little bit about Parr. Parr was born in 1893 in the northern region of Canada, Nunavut, where a lot of the Inuit people lived up in the Arctic. And he was a traditional hunter and trapper, so following the seasonal migration of animals for food and for sustenance. But late in life, he had an accident, and so he was forced to settle. And he settled here in what is Baffin Island, or now called the Kikitalik region, in the area of Cape Dorset, what was previously called Cape Dorset, but is now called Kingate. And so it was only really at the age of around 68 that Parr was encouraged to try drawing and to produce prints for what was then an emerging graphic arts program at Cape Dorset. And over the next 7 to 8 years, he produced some 2,000 prints, mainly black and white, although he did introduce some color later on, and really focused on this traditional way of life of hunting and trapping. And these drawings, although immediately in his time were not necessarily very well appreciated, have become a very, very important legacy, because they have provided this visual record of a way of life that was really transitioning in the 40s and 50s quite radically. And Parr was part of that transition. I mentioned the emerging printed graphic prints program at Cape Dorset. Now, this had really started in the late 1950s, and it really was, I would say, based and it required a more settled lifestyle, because if you can imagine, the Inuit as a traditional nomadic people were not carrying around a lot of materials. There was carvings going on, and there were some designs being produced on clothing and so on, but they did not have a way of life that would facilitate painting and drawing and so on. So once these settlements emerged in the Cape Dorset area, there was this emergence of a graphic arts program. And it was a very, very innovative program. Some of the techniques that they brought in to develop this form of graphic arts were adapted from Japanese woodblock printing techniques. And this was a very collaborative process, because you would have an artist such as Parr who would produce the drawing. You would then have a stone cutter who would incise who would overlay the drawing onto stone and cut away the negative portions or chisel them away. And there will be some interesting links, I think, between this and what Melina is going to talk about. And finally, there was the process of inking and pulling the prints. And although in some instances it was the same person who did all of this, in many instances it was different people involved. So it was a very collaborative program, and it has become over time very successful. It expanded to other regions. And so for this reason, PAR, as one of the first generation of these artists involved in this graphic arts program, is very important, as is the development of the program itself, because it gave us a very rich visual iconography and imagery of the Inuit way of life. So I'm going to move on now from the Arctic into Montreal to talk about an artist called Prudence Heward, who was painting in the 1920s and 30s and who was very associated with the modernist art movement. So I'll give you a few moments to look at this painting. So you'll see that this was painted Rosaire. It's called Rosaire, and it was painted in 1935 by Prudence Heward. Prudence Heward was born in 1896 in Montreal, as I mentioned. And I don't know, perhaps we can look in the chat later if some of you want to note some impressions that you have of Rosaire. But when I look at him, I can certainly sympathize. We've all been feeling very hot these days. And I think he looks like a very serious and stoic and solid boy, and hot and flushed in this white jacket that he's wearing, and certainly somewhat uncomfortable. And I mentioned that Prudence Heward was a modernist artist. So what exactly does that mean? One of the things that the modernist artists were exploring was an expressive use of color. So in other words, not just painting the exact color that's in front of them, but using different tones or different hues to bring out character or to bring out emotion in what they're painting. So your eyes may initially have been drawn to this very bright kind of pink orangey shirt that Rosaire is wearing. It's sort of echoed those tones around his nose. It's very flushed. And this tone really brings out the complementary blue color in his eyes, which we also see some blues behind here. We also have very solid lines and strong use of shadowing, for example, around his shoulder or light up the stairs and in the background as well. All of this to bring out Rosaire. Another feature that she gives us is we have the 3 stair lines, which all point to different elements of Rosaire that are interesting. We have this top stair, which points to his stair. I've got a little pun there. We've got the second stair, which really points us to this jacket. He seems to be really uncomfortable. And this third one, we see these hands, which are really very large. Prudence Heward often positioned people with folded hands. Again, an expressive liberty she took with making the hands very large. And of course, all this fluorescence coloring on his jacket that we see. I have brought up here a study for Rosaire that she did as part of this final work. And you'll see it says Interior Saint-Sauveur. For those of you that aren't from Montreal, Saint-Sauveur is the countryside north of Montreal. And Prudence Heward did do a lot of painting, sketching in the rural countryside. She was known for her portraiture, especially women, strong, sculpted women, often unsmiling, which was quite an unusual way to depict women. But often they were against a rural background. So Rosaire may well be perhaps one of the children of one of the local families. Perhaps the large hands may be a reference to farm work. Certainly he's an intriguing character because maybe he's 12 or 13 sitting there with his folded hands in his hot jacket. I'd be interested maybe if any of you have some ideas of what Rosaire is doing there at the bottom of the stairs, put those in the chat. I'd like to just briefly talk about Prudence Heward more broadly in terms of her background. She came from a privileged family. She was studying at the Art Association of Montreal, associated with our museum, where she was really encouraged by her teachers to experiment with new ways of doing art in terms of form and color. She studied in Paris, she spent time in London and traveled in Venice and other parts of Italy. And she would have been exposed to modernist painters in Europe, such as Matisse or Gauguin, Cézanne, and so on. And so she brought a lot of these ideas back to Montreal and advocated for their introduction into Montreal art circles, which were more conservative. And she was also affiliated with a group of Montreal-based artists called the Beaver Hall Group, which was an assembly of about 20 artists who shared studio space on Beaver Hall, which is a street in Montreal. Prudence Heward actually had her own studio on Peel Street, but she exhibited with them and was friends with many of them. And these artists did a lot of modernist art, portraiture, urban scenes, and so on. But one of the interesting things about them, and what was quite revolutionary at the time, was that almost half of the members of this group were professional women artists. And that in itself was also something within this time that was very unusual. And Prudence Heward, therefore, is really someone associated with new ways of doing things, not only because of the style that she was introducing and advocating for, but also for simply the fact that she was a successful female artist in the Montreal area within this Beaver Hall group, and more broadly in Canada as well. So that is our second work, and I'm now going to move on to something again quite different. I'd like to just briefly tell you what you're looking at, in the sense that what it actually is two circular canvases. They're very, very large. Each one of them is about 8.5 feet. So together it's a really monumental work, and they're on a gray wall. So it's not a painting of two, they're actually, it's called a diptych when you have these two circles. And again, maybe if some of you want in the chat to put your first impressions, we can look at those later. And also a question I have for you is, are these circles, do they have the same colors, or are they different? Are they the same or different? And this question of looking at color and figuring out if it's the same or different was really part of the exploration that Claude Toussignon was involved with from the outset of his career. So he was born in Montreal in 1932. This work of art, which is called 578-102, was painted in 1978, so he was well into his career. And he was an artist that had started in the abstract movement, but then moved abstract, but more spontaneous and free, but moved in the 1950s into what was called geometric abstraction. So a lot of exploration of form and color. And he was affiliated with a group of artists called les plasticiens. And they had these investigations of the plastic, it's called, elements of art, so form, color, and line, and how they can work together. And these artists really wanted to remove any subjectivity or storytelling from their art. They were interested in looking at the visual effects that you could achieve by putting different colors or different forms next to each other. And within especially the Montreal and Quebec area, Claude Toussignon, along with artists such as Guido Molinari, was really at the forefront of this exploration of geometric abstraction. So he initially, for example, did the circle was one of his signature motifs. He tried it on square canvases and on rectangular canvases. Then he moved on to circular canvases. He could use many, many different colors. In one of his paintings, he had, I think, 141 concentric circles in 7 different colors. So you can imagine that would be quite a vibrant work to look at. And he would have been influenced in his work by artists such as Joseph Albers, a German artist who came over to the United States in the 40s and became very influential and at the forefront of color theory. He did a series called Homage to a Square, where you had over 1,000 paintings purely looking at how different colors work together within the form of a square. And in Claude Toussignon's case, he worked very much with the circle. He also did sculptures and other formats, always abstract and always focused on this exploration of color and form. So these 3 artworks that we looked at are certainly all very different, but they all have an element of newness and new ideas. To briefly summarize, we looked at Parr's work, which was done in 1961, the stone cut engraving, really looking at how you can take a simple line drawing and construct a narrative out of it. It may be very different depending on who you are and where you come from, but there are certain visual cues we look at. And of course, in conjunction with the emergence of the graphic arts program, it really pushed a whole new form of art in the Inuit tradition. We have Prudence Heward with Rosaire, painted in 1935. So an artist who was really using, amongst other things, color and line to bring to the front a lot of expression and character in her portraiture. And we then have Claude Toussignon with the geometric abstraction, which seeks to remove any storytelling from his paintings and really get you just to focus on what we can experience by looking at color and different variations of color and how we can understand and be affected by color and form. So 3 very different artists, and I hope they've given you some ideas that you find interesting and that you will be able to link to what Melina is going to talk about next. So I will now hand over to Christian, I think, who will introduce Melina.
Christian?
Yes.
Can I read back just a few of the comments that we got in the chat about the different paintings? I think that Madeleine would find them interesting, and I think it'll be good fuel for questions when we go to. I'll just read them quickly. So, for your interest, Madeleine, we have of Rosaire. We have maybe he was posing for a family portrait on a spring day. Maybe he was in trouble and being punished. He's waiting for other members of his family to leave for mass. But perhaps he was being punished for posing for hours for being a pain, for being so young. And then when we go to Toussignon, we have eye test, a study of color relationships, 2 different colored targets. Reminds me of Joseph Albert. An optical illusion. Looks like they could be side by side the same color if they weren't. Oh no, looks like they could be the same color if they weren't side by side. They look different, but they're also the same. It's almost hypnotic. And then wondering if Le Duc and Toussignon were contemporaries. So there's a little food for thought of what came up in the chat. Thank you.
Thank you. Well, great comments, and maybe we'll talk a bit more about them at the end. I don't actually think I mentioned that the colors in the 2 circles are actually slightly different. But some people see that and some people don't, because it's just so subtle.
Perfect. Thank you. Thank you so much, Madeleine. So we will continue then with the second part of our colloquium, and then we will come back for all the questions and comments. So I would like to introduce, it's my great pleasure to introduce Melina Jaramillo Garcia, who is an imaging scientist at the Douglas Hospital Research Center, where she managed the molecular and cellular microscopy platform. So Melina completed her education in Concordia University, where she used advanced microscopy techniques to study cell division. Prior to that, prior to being now in the Douglas Institute, Melina was managing a core facility over there in microscopy, so helping people in the Department of Biology and teaching neuroscientists over there how to use advanced techniques of microscopy that you will see in her talk. You will really be awed by what she was showing us. So Melina is an expert in different techniques like epifluorescence, laser scanning confocal, multiphoton imaging to capture stunning images that you will see today. For her also is a special interest in scientific communication and the use of microscopy images to promote dialogue between researchers and the public. She has been helping also to organize events like images competitions and exhibitions for the users of the facility, and to make these images also public for the people outside of the institute. So this way, also creating a path for communication of science through images. Future projects include a collaboration between the molecular cellular microscopy platform, it's a long name, MCMP, with the Convergence Initiative. That this has been also one of these work that we have been cooking for years, and the pandemic shut down. So we are really hoping that this can soon that we can start to work with Melina to bring students to this amazing facility that is here in Montreal, so they can also enjoy producing images and then creating art that can talk about science in them. So I'm gonna leave now with the amazing Melina and her work, so you can enjoy it also as I do. Melina, all yours.
Thank you so much, Christian. Hello, everyone. It's a real pleasure to be here today. I've been looking forward to this talk for a long time. And as Christian said, I've been thinking about it for pre-pandemic times, you know, and then we got shut down for a year. So I've had a long time to rummage and think about what I was going to show you all. I was asked by Christian today to wow you. So, you know, to really show you the full range of rainbow colors that we work with under the microscope. And so that being said, I tried to sort of like tone down the heavy science of what it is that I do on a day-to-day basis and emphasize rather more the aesthetic part of my work. So I hope that, I'm sorry to the scientists, it's going to be a little bit you know, simplified for the scientists in the audience, but I hope that everyone will enjoy it nonetheless. As Christian said, I work here at the Douglas Hospital, and the Douglas Hospital is a McGill Teaching Institute. I run a microscopy platform. For those of you who don't really know what a microscopy platform is, you'll see a little bit of what it is, but essentially it's a facility where we house very advanced microscopy systems that are there to be shared and supported by the scientific community. So my job is to not only maintain this facility running smoothly on a day-to-day basis, but also to assist the students and to guide them in their imaging experiments to image their neurobiological samples. And that can be anything from all the way from sample preparation all the way to the publishing of their beautiful images. I will give a little disclaimer before I get started on the talk. So I am an imaging specialist. I am not a neuroscientist by training. However, I talk and I interact with neuroscientists every day for the last 5-6 years, so they do rub off on you. And after 5-6 years, I've acquired, you know, the basic rudimental knowledge that I need to basically talk a little bit about how fluorescence and fluorescence microscopy is applied in the field of neuroscience. But towards the end, if you have very, very deep questions about neuroscience, maybe I'll need a little bit of Christian's or Cecilia's help to help me along. Okay, so let's get started. I'm going to share my screen. Right here. Just a second. Share. So back here. Okay, so can everybody see this? Yes? No?
No,
No, no.
Okay, hold on a second.
No.
Let's see. Share screen. Okay. How about now?
Yep.
Okay, good. All right, so let's see. Perfect.
There you go.
So now it's good. Okay. Okay, so so let's get started right away. So I said, like I said before, I'm going to tone down the science, but I lied a little bit, and I'm taking you all back to high school physics. I swear it's going to be very short-lived. The pain will be very brief. So, you know, we were talking about colors and differences in colors, and one of the concepts that you need to really remember when you want to understand what fluorescence is that light and colors in the human visible spectrum can really be described as a wavelength. So as a wave. And so when we talk about colors in science, or specifically in imaging science, we don't often refer to them by their names. For example, blue. especially in fluorescence, we tend to refer to them by their wavelengths. So blue can be, or cyan can be something like 420 nanometers. Red can be 560 nanometers. Yellow can be, I don't know, 500 nanometers. So we really, I want to emphasize the fact that light and colors can really be described by their wavelengths. The cooler colors like blue, indigo, violet, etc., tend to have shorter wavelengths, around closer to the 400 nanometer range, so closer to the UV spectrum. And the warmer colors like red and yellow and orange tend to be closer to the infrared spectrum and have longer wavelengths, closer to 700 nanometers. So that being said, that will help us understand what the concept of fluorescence is. So the definition of fluorescence is really the phenomenon by which a substance, I don't know if when I move my mouse, you can see it on the screen or no?
Yes, yes.
Yes.
Okay, perfect. So the phenomenon of fluorescence is the phenomenon by which a substance, when it is excited with a specific wavelength of light, so a specific color of light, it absorbs that light and then it emits another color of light, another wavelength of light, usually and actually always higher wavelength of light than the excitation light. So there's an absorption part and an emission part. And so that is fluorescence in its essence, very simplified. There's two take-home messages here. One I already mentioned is the emission light is always higher in the spectrum, so higher wavelength if you want, than the excitation light for all substances that fluoresce. The second take-home message is that if I were, for example, the substance that is here gets excited with blue light, if I were to shine any other color light that is not in the excitation range of the blue light, so for example, red light, the substance would not fluoresce. So substances that fluoresce are usually very specific about what kind of excitation light they require to undergo that process. Of course, like I said, I simplified this grossly, and I'm not going to go through this schematic. I just wanted to show you how complex the process of fluorescence really is and how complicated it can be to harness it sometimes to do some imaging. So it's a physical process that even in fact some of it's still being studied. Imaging scientists like myself don't even fully understand it. And it's still being actively studied by labs of optical physics. So we talked a little bit about substances that fluoresce, and so I want to tell you a little bit more about fluorophores, which are basically what we call these substances. So fluorophores are really all around us. They're pretty ubiquitous in nature and in our environment. Very, very commonly known examples are corals. Minerals can fluoresce as well. And of course, you'll find fluorescence all over the animal kingdom and the plant kingdom. That being said, of course, this phenomenon has been observed for hundreds of years. And of course, scientists have always tried to understand it, but we've also tried to isolate the compounds from these natural sources. So isolate fluorophores in the lab and even synthesize our own in the lab for an array of uses. It could be for biological purposes, but also, the industry. So everybody knows about your highlighters, your fluorescent highlighters, so these compounds are fluorescent and have been synthesized in labs for, decades. So, but what's important to note is that after so many decades of studying fluorophores and, isolating them in labs and synthesizing our own, we have a full range of palettes of these compounds to play with. They not only range in colors and in their excitation properties and their emission properties, but they also range in other physical properties, for example, or chemical properties, for example, and more specifically, they range in their ability to interact with biological molecules, and this will become important later on. So, like I said, it's been studied for a long time, but I wanted to tell you a little bit about the story of the jellyfish and the green fluorescent protein, because this is one of the most successful stories and really what pushed fluorescence microscopy to the forefront of biology and medicine. So in the 1960s, scientists in Japan isolated the fluorophore that makes Aquaria victoria, this beautiful jellyfish, fluoresce. And the fluorophore is an actual protein that is, produced by this organism. And this protein is called green fluorescent protein, or commonly known as GFP. Over the subsequent decades, GFP was, characterized, and both its DNA sequence and also its structure its inherent structure. You can see it here on the left of the screen, a little animation of what GFP actually looks like. It's basically like a barrel, and at its core, it houses the fluorophore that allows it to fluoresce. Where the innovation and the giant scientific leap truly happened is when, in the 1990s, a group decided to take the DNA sequence of GFP and clone it into another organism to use it as to use it basically as we call in science a tag or a reporter gene. So what that means essentially is that you have a protein of interest. The DNA sequence of that protein of interest is somewhere along the genome. You insert right after it the DNA sequence of GFP. And so when your organism and the machinery of the cell produces this protein of interest, it will produce it with this little bright beacon attached to it. So you will be able to follow the functions and the interactions of these proteins sometimes in real time, everywhere around the organism and the cell, most particularly. So that was a huge leap in science. And so you can see here different organisms that have had GFP cloned into their genomes. The first one was the worm. So you can see here the C. elegans worms. But it quickly, the technique quickly spread all across biology to encompass anything from fruit flies, zebrafish, mice, plants. I mean, it became pretty much a staple of biological studies. Not only that, but because we knew its DNA sequence, some labs dedicated themselves entirely to engineering variations of GFP. And so you can see here at the bottom of the screen that, these are just a few examples. Nowadays, there are hundreds and hundreds of variations of GFP and different fluorescent proteins that you can use as a tool. But basically here you can see blue fluorescence protein, so EBFP, CFP for cyan, GFP, YFP for yellow. And then the names become a little bit bonkers, like honeydew, banana, orange, cherry. That's when the scientists start to, get creative and their inner artists come out. So what I really want you to understand at this point is that we've developed an entire palette of fluorescent proteins so that we can clone these into different organisms and watch their interactions in the cell at the cellular level. You can see here on the right next to the fluorescent protein engineers on the side, you can see a Petri dish that has been smeared with bacteria. Each color of every single line of smear of bacteria is a bacteria that has had cloned a different fluorescent protein in it. And so whoever made this beautiful Petri dish decided to get quite artistic on that day. So now we talked about the fluorophores. We talked about the cloning of the fluorescent proteins. And so you can see here again, green fluorescent protein in a tobacco leaf. That's a great model. Simple fluorescent compounds. I said before that fluorescent compounds can have interactions with biological molecules. I'll give you some important examples. For example, the molecule DAPI, which is widely used in biology, is a molecule that fluoresces in the blue spectrum of the visible spectra, and it attaches itself to nucleic acids, so to DNA and RNA. So when we want to visualize nuclei in cells, so you can see the cells here, and when we excite DAPI and we look at DAPI under the microscope at the right wavelength, we can see all the nuclei under the microscope. We can also use things like phalloidin, for example. That's another example. Phalloidin attaches itself to actin, which is basically a scaffolding structural protein that is found in all cells. So you can see here the fibers of actin holding the cell shaped together, all stained with phalloidin, which in that particular case fluoresces in the red spectrum of the visible spectrum. So those are two, staining techniques that we use. So one is cloning fluorescent proteins directly into the genomes. One is just staining directly biological compounds with fluorescent chemicals. But the most powerful technique, and I would say the most commonly used technique in the lab to stain or paint our cells under the scope, is called immunofluorescence. And so immunofluorescence, the word says it's immunology. So it basically uses our knowledge of antibodies and uses that to target very, very specific proteins of interest in our cells. So if you look here, you have a cell, you have a target of interest. We develop antibodies that will target very specifically that target, but attached to these antibodies will be fluorescent compounds. And so the power in this technique is that it's so specific. So if I wanted to look at something that is very, very unique in a cell, I can do this. And I can do this once, but then I can use another antibody, attach another fluorophore to it, and then so on and so on, create layers of colors within my cells. And you can see here at the bottom, some cells that have been immunostained for a protein called tubulin that is also a scaffolding protein in the cell. So now, we have not only a palette of colors, we have different techniques of labeling with these colors, but none of that is useful. None of it is useful if we don't have the means to actually capture that little fluorescent signal once it is emitted. When an animal, or the tobacco plant that you just saw, or the fluorescent bunny is fluorescing, you can see it to the naked eye. But biologists are interested in what's going on at the cellular level. So it's very important that we combine fluorescence and fluorescent labeling techniques with very advanced microscopy. So when I say advanced microscopy, I'm no longer talking about the little tabletop microscopes that you have in Biology 101 class or in high school, whatever. I'm talking about very, very advanced systems that use light sources like lasers, very sensitive cameras and detectors, and of course, very powerful objectives, as you can see here. It's funny because when I usually give talks about fluorescence microscopy, 90% of my talk is explaining that stuff, what's going on here. But today, it's the opposite. I'm rather explaining what's going on at the color level and not at the technical level of the microscopes. So we've talked about the fluorophore palettes, the labeling techniques, and the advanced microscopes. So when you combine all of this, the possibilities are truly endless. So at this point in the talk, so this is the half point of my talk, I want to tell, I've told you all about the techniques and all that stuff. So now it's more like an exhibition. I'm going to show you some of the applications of fluorescent microscopy into neuroscience. We were joking about this yesterday. This would have been so good if we were in a dark auditorium. And so if you have a dark place to go in your home, and if you want to turn off the light, this is the perfect way to visualize these images. So instead of going to your happy place, I'm going to ask you to go to your dark place today. So this first image here is the image of a hippocampus. So in a mouse brain slice. So this is a classic image in neuroscience. You know, the hippocampus, it's studied quite a lot. It's involved in memory. We know that it's involved in memory. And in this particular image, you can see that DAPI was used to stain the nuclei. So you can see in blue that DAPI was stained the nuclei. And then you can see this mouse, before it was sacrificed, was a transgenic mouse inherently. So it was expressing GFP as part of its, normal life. And it was tagged to a protein called CFOS that is expressed in very particular neurons. And so CFOS, GFP tagged CFOS neurons are seen in green along this layer of the hippocampus. And in red, what you see is a derivative. So some neurons that are called OLM interneurons, and these have been stained in a different way by another fluorescent protein that's called TD tomato. So it's a derivative of a red fluorescent protein. And the way the TD tomato was expressed in these animals is it was injected in a virus. So the virus machinery of hijacking into our cells was used to insert the TD tomato DNA sequence into the mouse, allowing the mouse to express TD tomato alongside the OLM interneurons. So that's what you see here. It's a beautiful image that was acquired by students in the Williams lab here at the DHRC. The next one, and this is one of my favorites. So Nguyen, one of my users in the Ernst lab here, acquired this beautiful image using a laser scanning confocal microscope. What you see here is basically a brain in a dish. So I'll explain what I mean by that. Most of the time when you are studying psychiatric disorders, or neurodegenerative diseases, you have models that you can use. So, for example, mice that have Alzheimer's, or you have access to, for example, human postmortem samples that you can study under the microscope. But once in a while, you'll have neurodegenerative diseases or people who are born with them that are so rare that there are no such models. And so what the Ernst lab has done, and what they have come up with to circumvent this, is they basically collect stem cells from the urine of these young patients, and they reprogram them in cultures to basically behave like neurons. And so when these reprogrammed cells are in the dish, they start to make connections just like they would in the brain, and they start to organize themselves into masses that we call organoids. And so this is what you see there. That mass that you see there is an organoid. You see that DAPI, again, is used to stain the nuclei. And then you have immunolabeling of TUJ, which is a structural protein, and MAP2, which is a protein of interest that is being studied for this particular neurodegenerative disease. So I really love this image. It almost looks like, a supernova, or like two planets, a planet and a moon circling each other, Okay, so next one. This is a human postmortem sample showing and staining what we call the region of the brain that's called the dentate gyrus. Claudia is one of my most enthusiastic and brilliant microscopy users here at the DHRC. She works in the Meshawar lab, and this lab is interested in the adverse effects of childhood abuse into adults that suffer from depression and commit suicide. And so these samples come actually from donations from patients from the brain bank. And so she has immunostained this sample. Again, DAPI, it's ubiquitously used in neuroscience to stain all the little nuclei. Then she used Alexa488, which is an antibody that we use very, very commonly in neuroscience or across all biology, really, to stain neurons expressing nous. And then in red, so the little red dots here are perineuronal nets, and perineuronal nets are what Claudia was really truly interested in this particular study. So one last one before I go into the next one. So I've shown you so far, things in a dish and tissue, pretty wide tissue. But once in a while, we really have to go to magnify and go and look at things very, very, very closely. And so what you see here is an image from the Flores lab in which you see just a little branch of a neuron, of a pyramidal neuron called a dendrite. And along these branches, you see where the little green arrows are pointing, these little protrusions. Oops. You see these little protrusions that are called spines, and spines are, thought to have a variety of roles, memory included. And so the study, these images are in fact used to record the density of the spines along the dendrites, but also to classify their shapes. So they have different shapes, mushroom, stubby, they have all these funny names. And so the idea behind these images is eventually to classify all of these, characterize all of these little spines along the way. So, like I said, the possibilities are really truly endless, but it doesn't really stop there. In fact, if you start to modify and tweak at these techniques, you can even get more possibilities. And now I'm really going to go into what I call truly advanced imaging. So this is even advanced for me. And, when I get to play with these kinds of samples, this is like a kid in a candy store. So the video that I'm going to show you is a video of a zebrafish embryo. It was not acquired here at the DHRC. It was acquired by a colleague of mine, Elizabeth Haynes, and Henry Hay at the University of Wisconsin. This video you can watch on YouTube, as much as you want. It was the winner of the 2018 Nikon Small World in Motion competition, and you will see why. The image is in black and white, but the zebrafish is expressed in GFP, in basically tagging a protein that is expressed in the sensory neurons of its nervous system. What's really important to notice here, the innovation is not in so much that the zebrafish is a transgenic GFP fish that has been around for a long time. It's the microscope that was used. The microscope that was used is a very special microscope called a spin microscope. And so that's where the innovation comes in. The spin microscope is a very gentle microscope, but also a very fast imaging microscope, and it allows you to image with very, very high sharp resolution. And so you will see now the development of this beautiful zebrafish embryo. So what you can see along the way here are these sensory neurons that are developing here along the spine. And then as the development continues, you can see the neurons growing these axons on the side of the fish, of the body of the fish. So this would be the head, this would be its future body. And so these sensory neurons will be what will allow the fish to sense touch really on its skin. Beautiful video. I love it. I watch it on the regular. Wish I had some zebrafish to play with at the DHRC, but I don't. So next, another innovation. We use GFP, as I said, as a biological tracer, as a tag. So when you have no GFP, your protein expresses normally. When you insert the sequence of DNA of GFP or any other fluorescent protein for that matter, you express the same protein, but now it has this little beacon attached to it. So that's how we use GFP or fluorescence proteins as a tag. But, oops, sorry. But what if we decided to insert something a little bit more complex ahead of the gene of interest? So these DNA sequences that we can insert ahead of our genes, ahead of the gene of interest. Oops. Okay, I don't know what happened here. Okay, there we go. This one, so this one is called a Cree recombinant box. And so basically it has a series of fluorescent protein DNA sequences ahead of it. And so if you combine a couple of boxes together, so 1, 2, and 3, ahead of your gene of interest, what the Cree recombinant box does is it randomly, in each cell, it would randomly excise and remove some of these colors out of the way. So in cell number 1, for example, it may remove everything but the blue. And in cell number 2, it will keep a little bit of the red. Or in cell number 3, will only keep a bit of yellow and a bit of blue. And so, as you can imagine, what you start to do is genetically give a blueprint to basically express from cell to cell a different palette of color. And so this process is completely random. And so this is how you achieve what we call the brainbow. And the brainbow, I mean, it's absolutely visually stunning, but it also has a very important biological application in the fact that if these neurons, who are very, very densely packed in the dentate gyrus, were to all express just one color, it would be very difficult for scientists to sort of make out their connections to each other, because everything would be green or one single color. And so the brainbow technique, or the multi-color genetic labeling techniques, is basically a technique that allows us to really see the connectivity between different neurons in the brain. And so people who are studying what we call the connectome in the brain can really, really start to see different connections and how they are made from neuron to neuron. So I hope you appreciate how visually stunning this is. And it's been, Brainbow in particular has been the inspiration for many, many artistic endeavors by scientists and whatnot. And you can rightly see so why. Okay, so last example of a very important innovation in neuroscience. In this particular case, it's a combination of an innovation in microscopy and a technique of sample preparation. Oftentimes, we cannot see brains fully in 3D. Why? Because brains tend to be very opaque and they are very fatty. And the problem with something being fatty is that fat itself absorbs a lot of fluorescent light. And so it becomes very difficult to observe our brains very deep into the tissue. And so what scientists do is they slice the brain into very, very thin slices usually, and then they image each of these slices along. And then later on, with very complex software, they reconstruct the brain together. But what if we could remove what it is that makes the brain opaque, so all the fatty lipids that makes the brain opaque, and just keep everything else in place, including all the reporter markers like GFP, etc. And so this technique is called clarification, and you can see here how you can go from a very opaque brain to a completely transparent. I mean, you can't really see the brain, but you can see that you can read through it. So the transgenic mouse is then, is all intact. Everything but the fat of the brain is kept. And so and then we image this with a special microscope called a light sheet microscope, which is essentially a microscope where you can image large volumes very, very quickly. It's nice to have a clarified brain, but if you don't have the microscope to image it, it could take you a week or so to image an entire mouse brain or a rat brain in high detail. But with a light sheet microscope, that technique became quite popular because suddenly we could image a whole brain within an hour. So I'm going to show you here what a clarified 3D brain looks like. This brain is showing Th positive neurons, so neurons that have dopaminergic neurons. And so you can see how powerful this type of imaging can be because you can see immediately the 3D anatomy of the mouse brain and the distribution of this signal across different parts of the brain. Not only that, but if you have the proper light sheet microscope, you can image it in such detail that you can travel to the brain and see every single little neuron across all the connections, and it's truly marvelous to be able to 3D virtually travel through these kind of images. In fact, 3D light sheet microscopy of clarified brain has given us ideas about how we interpret data, really. So we usually show these kind of data in journals in 2D or in presentations like this, but now we are starting to show these data through virtual reality. So imaging the brains in 3D has allowed us to do that now, and the way we interpret now this data is completely different than it used to be. Okay, so that was it for me. I want to conclude by saying that I really hope that that you saw how wide the range of fluorescent microscopy can be, and the wonderful things that we can do with it in the field of neuroscience, and that these images, I consider them to be works of art in and of themselves, but they are often the inspiration for more traditional works of art, like, for example, this piece that's called Starlight Gone, that was created by Liam O'Leary and Elizabeth Parent through the Convergence course at Concordia that brings neuroscience students from McGill and art students from Concordia together to create these wonderful pieces. And so Liam was one of my most brilliant and enthusiastic microscopy users at the DHRC. And this particular piece was created using some of the data that he had acquired through his PhD in the Meshawar lab. So I hope that it can show you how microscopy data can be used to communicate science to the public, and make the public aware of the wonderful research that we do in neuroscience. So that's it for me. Thank you so much. I hope you like it and enjoyed it.
Thank you so much, Melina. That was a really, really stunning presentation. Amazing.
Thank you.
So let's bring everybody here now. So just need to, there you go. So hi, everybody. So now we really can start, to talk about this, like all these amazing images and techniques. I think it's just wonderful how we can see in both realms, right? Like how the advance of technology can move the creativity to just achieve new places. So let's start maybe with what we were talking before with Madeleine. So some people was making comments about the paintings that you were asking before. So Stefan, you want to take here?
With pleasure, with pleasure. So as we do with all our colloquiums, we invite you either to use the chat or we'd be very happy to see your face if you want to open your microphone. Do you have some questions for either, both of our panelists? Do you have some reflections? We've gotten comments like mind's blown. What is it that's come up for you that you would like to know more about that maybe we can help answer? When we were looking at a visit for the fine arts, we were really looking at what happens in those moments of innovation. And in the artworks that we were looking at, we had artists that were confronted with different impediments to their own human advancement, whether you could no longer perform your way of life traditionally, whether your gender was impeding you from moving forward according to how society perceived you, or that you were exploring a frontier of representation. So if you recall the artworks that we were looking for, do you have some more questions about what you saw that we can help answer? And then let's also go over to our neuroscience side of things, that we've stepped into this fantastic molecular world of fluorescence. What came up for you that you would like to know more about? So let's see what people are putting into the chat. We already have some questions to our neuroscientists, but I personally have a question for Madeleine, if I can jump right in and ask you, because I personally love when there are these critical moments. The most exciting moments in art history is when something is reacting to something. It's often how we find ourselves trying to explain abstraction. How did this come about? Why is there a reduction? Or why is there an addition? Or how did this come out of something that didn't previously exist? In the examples that we looked at, each of them which are a departure either personally or within the art world, what for you was the most personally intriguing moment of crises that produced this innovation? You look at the artworks that you presented.
Well, I certainly found looking at Parr's work and the development of the graphic arts program was very interesting, because when I was researching the artwork, I really learned more about, and this is really at a very top line level, I'd like to say, just some of the disruption that was caused by the changes that were forced or happened to the traditional way of life and the move into settlements, and how that led to looking for ways to provide a means of living to people whose lives had been quite radically changed. So there was the art portion of it, and I love those prints, but just understanding more about the context of it. And in the Prudence Heward work, I found it really made me think a lot about the changes that Prudence Heward. She actually went to London in 1914. One of her brothers was in the First World War, and she served with the Red Cross there. But during the First World War, there were a lot of changes in society for women. The women that were back home, they took on new roles. And so there's a real evolution and changes in fashion and so on. So that kind of turning point, really looking at the Prudence Heward work, made me think about it more just in the context of an artist, and how that can affect them, and the barriers that push through. But there was that external context in both cases that helped shape some of the things that led to the artworks that they produced.
I was particularly intrigued by the out of norm representation of the figure, and unsmiling, for instance. That just it immediately struck me. But also the use of color was really unusual as well. We hear a lot about Beaver Hall alongside of what's happening with the Group of Seven, and we usually sort of tend to think of it as the Group of Seven really helped to usher in Beaver Hall, but we don't hear enough about how Beaver Hall really influenced the Group of Seven, and how along those gender lines there really was this radical departure in terms of how women artists were invited, permitted even to have a voice and to change the direction of art as we know it. I really learned something there. Oh, Christian, I see a hand.
Actually, that made me think a lot. I was just this weekend in the Musée des Beaux-Arts du Québec. And in terms of representation, I was just surprised at how bad the representation is in the museum. Like, I didn't see, there's 4 beautiful pavilions dedicated to 4 male artists. One of them, I actually have questions to you about the work of Toussignac, if he was in a relation with Leduc. We also was playing with colors. But referring to what Stefan was saying here, there was no woman pavilions, and it's not that Quebec or Canada doesn't have women's, right? Like to my mind, I always come the Beaver Hall. We have like an amazing production of amazing painters by women, and there is nothing dedicated to them as you see for guys. So there were 4 pavilions, one huge room for each one of them with amazing paintings. But as there have these amazing paintings, there's also amazing paintings for Canadians and Quebecers painters who are women. So I think it's a lot of work to do, and I want to use this stage today, because it's our last stage where we have public and all that, to just make this, a conscious call. Let's remember that 50% of the human beings are women, the other 50% are guys. So it should be the same representative thing in everything that we do. Anyway, that's say.
Thank you. I just wanted to mention, because you mentioned Fernand Leduc, and I don't know very much about him as an artist, but he was a part of the automatist group, which preceded the geometric abstraction with the more spontaneous expression. And then I believe he did join the first group of plasticiens, the ones that were more moving towards the more hard edge geometric abstraction in 1955. I'm not sure if he was a signatory of their manifesto, but I believe he did make that transition into this sort of new second wave of abstract painting. Perhaps one of the other guys or somebody else knows a little bit more about him, but he did, I think, have a foot in both camps.
That's so cool. When you were mentioning also that Toussaint was influenced by Albers, and I was thinking Albers the moment that you put the 2 circles there, and I just love how all these people is connected. And then when you have this opportunity to visit museums and you remember, oh, this guy remember me this other one. Mm-hmm. And then you have now the knowledge that, yeah, they actually knew each other. So they were influenced each other by their art. Mm-hmm. So just showing also that sometimes for us, commoners outside of the art, we think that artists just come with this from their brain, but no, it's influenced in everything that they do as it happens in science.
Yes, indeed. Yeah.
Christian, would you like to share Nora's question that we have in the chat?
Yeah, sure. Nora, do you want to do that with your brain? Okay. So Nora, jump and ask your question, please.
Yes. So I'm curious whether I could be the guinea pig and do a study on like biopsy. That sounds so intense when I say it out loud. Can I do this on a live person like myself, do this imagery, or is it exclusive to already deceased beings? How do you logistically do this?
Okay, that's a really good question, actually, and something that scientists are trying to circumvent and get closer and closer to, in fact. But as of now, except for MRI imaging, which is very common in neuro, medicine and whatnot, when it comes to fluorescence microscopy, to be able to image within the brain, you got to open the skull. So no, the answer, the quick answer is no, we do not do this on live people.
I don't think my Concordia health insurance is going to cover the cost of that.
Exactly. But that's why we have animal models usually. And so in animals, we do this, and we've done this here at the DHRC a long time ago, where we perform a surgery on, actually, Cecilia is very familiar with this. We perform a surgery on a mouse, open the skull, a little square on the skull, and we insert what we call a Grin lens. So like a little, objective lens. And this objective lens is connected to the microscope, and you can image the neurons firing, and giving off those GFP signals and those fluorescent protein signals, usually using a fluorescent protein that we call GCAMP, which is a derivative of GFP, but responds to calcium signals, which are necessary for influx between neurons. And so when you can do this while the mouse is still alive, usually immobilized. But recently, in the last couple of years, we have done this. The microscopes have been reduced to such tiny, tiny portable microscopes that now we don't have to restrain the mouse from moving along. We can implant the microscope directly on top of it. They're called miniscopes. I don't work with miniscopes. Cecilia actually works with miniscopes quite a lot. But the miniscopes are essentially tiny microscopes that allow you to image how the neurons are firing while you give these animals a task to do, memory retrieval, adverse effects, and all kinds of stuff. I mean, it's incredible the amount of innovation that it takes to do something like that. To take a microscope that takes half a room and put it into the palm of your hand is an incredible technological feat. So yeah, it is possible, but not in humans yet. When it comes to humans, most of the samples that are imaged using fluorescence microscopy are human postmortem samples from people who have donated their brain to science, or their families have donated their brains to science. In fact, here at the Douglas, we have one of the most famous brain banks in the world. It specializes in depression, mood disorders, and suicide. So that's why you will hear researchers from the Douglas are very specialized in these psychiatric disorders, because we have access to these samples on the regular. And so it's a really big privilege actually to have these samples under the microscope. And we're all the time very grateful to the people who donate these incredible samples to science.
That's awesome. Thank you so much. Very inspiring. Thank you.
Nora, I wanted to add here, because here it's very interesting what you asked, because each technique always push our knowledge, but also push the ethics of how we apply them. And this happens also in every field, as it happens in arts also. And in arts, actually, the fact that science has pushed these techniques to being able to modify things, to put things in animals and change colors and things like that, has created a line of artists that do biohacking. And biohacking is what you are asking. I'm not recommending it to you, just for the record. I'm not saying that you should do it, but some artists have arrived to the point where they injected different type of genes with different type of techniques and modifying their own bodies. Now, that is for a whole subject and discussion in bioethics and ethics. We will not go here in that. We will do it probably in the class. So, Stefan, all yours.
Okay, thank you. I'm looking forward.
Thank you, Christian. I see a hand, but first I want to acknowledge that Marie-Josée had her hand raised. If you still have a question or a comment, I'd be happy to go to you.
No, I didn't have my hand raised. I just move around.
I do too. I just wanted to acknowledge the gesture in case you had a question.
Very interesting.
Great, thank you. I do see the hand of Andrea. Would you like to open your mic and ask your question or offer your comment?
Yes, it's a question for Melina. Just out of interest, is the Douglas involved with the study of brains from sports injuries, which have made the headlines in the past decade? Of course, your hockey players, football players, soccer players, etc. And I suppose the imaging that you're doing would be extremely useful in that study.
So I'm not aware at the Douglas that anybody is doing that study. If it is being done, it was probably at the MNI, because the MNI has an expertise in brain trauma and injury. But it doesn't mean that they may not be using these techniques that I'm talking about in relation to that study. In fact, Thomas Stroh is my counterpart at the MNI, also runs a microscopy platform there, is a professor also at the MNI, and would probably know a lot more about that. But they also have the access to the same advanced microscopy systems that we do here at the Douglas. And so if ever that study was being conducted there, for sure. And I can easily see a study, in animal models where you have traumatic brain injury or something like that, or, postmortem samples that you can study and stain in the same way we do for suicide patients, and see if there are any cellular changes at the cellular level or connections that are being broken when somebody goes through traumatic brain injury. Yeah.
Thank you.
We're studying the lab traumatic brain injury in mice model. And the Montreal General also have a vision lab dedicated to that. They also the study traumatic brain injury in what happened in vision. And as you say, Melina, in the MNI also they have like a big group of people working. So it's a very, very interesting subject, particularly in Canada, because hockey players, because all the damage that they have received during the games. So I have here a question from Trish. Trish was asking to Melina for the name of the microscope that is used for clarity.
So the name of the microscope we use for clarity is usually 90% of the time it's called a light sheet microscope. And the reason why we call that a light sheet microscope is because the lasers that are being used, I don't know if you saw in the image, you had these lasers coming in at an angle like this from all directions. Usually lasers are very, very thin beamed, more like a thread. But in this particular kind of microscope, what we do is we modify the laser beam to be sort of like a flat sheet. So more like this rather than like this. And so this allows us to sweep larger volumes much faster. And so if you're doing it with one light sheet, that's great. If you're doing it with 6, like you saw in that image, then it's going to take six times less the amount of time to image a whole brain that has been clarified. It's called a light sheet microscope. Yeah.
And this is potentially a question for Melina or any of the researchers that we might have on the call about postmortem study into Lyme disease and its effect on brain and behavior. Does anyone have any information on whether that's being studied or not?
I don't know. Lyme disease, you said Lyme disease, right?
Lyme disease, yes, exactly. Yeah, Maureen is interested in the tick-borne illness. Mm-hmm.
Yeah, so I'm not familiar with Lyme disease research myself. Like I said, Douglas is very niche. It's psychiatric and mood disorders, and I'm a bit familiar with what goes on at the MNI a little bit also, but I'm not sure anybody. Christian, maybe you know anybody?
Maybe some people, maybe some people in the Glen. The Glen Hospital has an institute that study tropical diseases, and sometimes just because tropical, they classify exotic, and then Lyme may be there, but I'm not sure. I know it's a lab for malaria also, and then maybe there is some connection to Lyme, but I'm sure someone is doing it because it's prevalent in Canada right now and it's a big problem. So, but not 100% sure. Just put in Google, what I recommend is just put in Google Lyme, brain, and Montreal research. And then you will get someone here or in the Montreal, in the Université de Montréal also that will be working in that. If not, just send us an email and I will check for that. You just need to remind me that and I can check for that if you need it, and I will look for the information and let you know.
One thing I'd like to point out though, so just to relate to this question, any disease, any field of medicine, you can be sure that they have access to a microscopy platform just like mine, and that they are putting their samples under fluorescent imaging, and they are doing fluorescent imaging or some kind of imaging, advanced imaging, to study the cellular processes of these cells that are diseased. It's so ubiquitous in the field of medicine and biology. It's everywhere. If you are in a group that studies medicine in any kind or biology of any kind, it's very unlikely that you haven't been in front of one of those microscopes.
So I'm a psychotherapist, and I'm definitely interested in the research you're doing and the impacts that it may have on finding markers related to depression, suicide, etc. But my question for you is actually so much more as a neophyte. I remember the first time I saw an image of the human connectome, And if this is something new to anybody on the call, maybe it's not new to anybody on the call, but to me, it was something fabulous and wild and new. And when we're done with the call, please Google it. Just the human connectome is so, the image that you showed us was gorgeous. And it's just, my question is, and I don't know if you can answer it, my question is, this is mapping, right? That we're invested in the finite world of the mapping of all of these neurological connections. And I think that it's probably teaching us fabulous new things about how the brain actually works, because we still know so little about it. Any idea how far along we are in this mapping?
Oh my goodness. So we are, I mean, had you asked me this a couple of years back when light sheet microscopes and spin microscopes and all these new microscopes that are being developed at the speed of light, like at the pace of it, I mean, even I sometimes can't keep up. I have to go to conferences every 2 years to be able to keep up with the technology. But in the last 5 or 6 years, there have been such a huge leap in 3D imaging of the brain that I have to say, I'm not sure how far we are, but we've made huge strides in the last, I would say, I don't know, Christian, maybe like 7 years, I would say, in the human connectome, because before that, it was difficult to image the brain in 3D. So we could do it through MRI, but MRI does not allow us to specifically target, certain proteins or certain neurons very specifically. Whereas fluorescence microscopy does that, or transgenic animals does that, but we have to be able to image them in 3D. And so until light sheet microscopes and the lattice light sheet microscope particularly, this is a microscope I didn't show today because it's one of the most advanced ones. It's very hard to even work with one in Montreal at the moment, but these microscopes have allowed us to really, really image the brain in such detail. So prior to that, you could do 3D imaging with light sheets, but you couldn't zoom in detail and still retain a beautiful image. Now you can. Now you can, with lattice light sheet microscopes, not only can you do a full sweep of the brain, but you can image so, like you can zoom in so close to the cells that mapping a connectome is getting very, very, very close indeed, I would say.
Now, I just want to add to that it is important to understand also that many of the techniques that are used today are done in brains that are fixed, meaning they are dead, and then you just stop them in time, and then you visualize them. But now there's also techniques appearing more and more where you can do these connections in functional tissues or big pieces of tissues, where now you can do exactly the same thing, like connect one neuron, know if exactly that neuron is really connecting to another one and not just by physical distance. That's what happened, what you see sometimes in Brainbow, right? Like you see the connections there, but you need more evidence to know that they're really interacting. So now you can arrive now to the point where you can see interactions in real time in pieces of tissue. Not a complete brain like as you see in Clarity, but we're moving to that area now. So that's also important to mention.
'40s, I think it was, when the paint tubes were invented. It was transformative because it allowed the painters who had been really having to paint in their studios to go outside. And that started the whole tradition of plein air painting with the Impressionists and moving towards working more with light effects and so on, and changes of light outside. So I think it was really fun in the two presentations and watching Melina's presentation as well to look at how in the science labs they work with the colors to get the contrast right. And it brought me back to thinking as well about Joseph Albers and his squares and working a way to look at those at those differences. So as I find there's just so many parallels in so many different ways.
That was actually the one question I have for you. Like how you see all these techniques that Melina is talking today affecting and influencing art to now go to new exploration. How you just elucubrate, what do you think?
Well, as I said, I think, you know, in all fields you see things moving, but I think what particularly struck me, Melina, was especially with computer technologies and the art that's being produced, you know, 3D or otherwise with these new technologies. And that last image that you showed us of, I think it was one of your students who had worked within the Convergence Initiative to produce this artwork inspired by science, but sometimes it's just the image itself of somebody's brain or a mouse brain that becomes a work of art. So where does the line become when we look at it? I guess that's sort of one of the questions I have in my mind, because both images, whether they were conceived of as art or whether it was an image of a scientific study, in some ways they really weren't very different if you're just looking at them. They were just so beautiful.
Can I say something about that? So I was asked that question in the interview a couple of weeks back. How do you draw the line? Like, where's the line? And for me, because a scientific image can carry emotion, for me, there is no line as long as you have some sort of emotional connection to that image. And I was telling, I don't know if any of you read the interview, but I was telling Rebecca, who interviewed me, she asked me this, and I said, you know, I have students sitting at the microscope sometimes for the first time. They have toiled away for weeks preparing these samples, you know, growing their mice, taking care of their mice, preparing these samples. And they come to the microscope and they don't know if it's going to work, if whatever. And I often have students cry when they see their images and how beautiful they are. There is an emotional connection there, just like any other piece of artwork, you know. An interpretation as well, whether it's scientific, but also human, you know, at the human level. And I see this particularly with people who are working with post-mortem human samples, because we are working in a field where emotion is predominant. We're in a mental health institute where we study depression and we study suicide. So there is some sort of, like, you know, emotional connection there that happens that makes me think that these images are not just images of the brain. They do have an artistic and human component to them that dissolves that line, you know, that you're talking about.
Yeah, that's definitely food for thought. Very, very interesting and very true. I find that a wonderful expression of how to look at it.
Which is why I'm so excited to have artists from the Convergence here, because we're not only a science research center, but we study something that is very emotional. And so I'm hoping that when artists come to see me here and work with the samples and work with the microscopes, that they can harness that emotional connection and into their artwork, you know?
We will go there very, very soon.
I hope so. I hope so.
Let's continue pushing vaccination on people, and then we will be there.
Yeah, we're almost there, Christian. We're almost there.
Nidia, so
yes. Hi, Mom. This is Mom. Everybody, welcome. Okay, Mom, you have to remove your microphone.
Okay. Congratulations, a very good presentation. But I have a question, but I don't know how to say in English. Melina can traduce maybe.
Yeah, me and Christian will translate, no worries.
Melina, el cerebro tiene una enfermedad, esos colores fosforescentes, cuando se ven en el microscopio, cambian. Si por lo menos una molécula está enferma, ¿va a cambiar de color fosforescente?
Okay, that's an excellent question. So my mom is asking, she's asking, when when you are seeing samples that are diseased and stained with fluorescence, does the fluorescence change in your sample? So does the color change or is modified anyway? And the answer is yes. So most of the time, what you see is an on and off kind of effect. So you can have a control person that is healthy and expressing a particular protein, and you see the fluorescence, and then you'll have a diseased person, and then you don't see that fluorescence in that sample. Another thing that can happen, you can see in a control person a certain level of fluorescence, and then in a diseased person, an overexpression in which you see way brighter levels of fluorescence. Another particular thing that can happen, and this is particularly true in neuroscience, is how cells interact with each other. So sometimes the fluorescence doesn't change, but now it's somewhere else in the cell or interacted with something else. So what fluorescence allows us to do is to track not only location, but also function, the function of these proteins and these targets in the brain, and the connections that they have with other neighboring cells and whatnot. So yeah, good question, Mom.
That was a great question. Muy buena pregunta, Nidia. Buenísima.
Gracias, gracias.
So, is anyone that have any other questions? So, because it's already 7:10, so we have exhausted our speakers. So, if no one else have an immediate question, we will thanks to everyone for being here today. And again, thanks for participating in this colloquium. This is our last colloquium for this year. We are working already with Stefan, his team, and our team to go with a new subject for next year, hopefully. And just remember, next Saturday, Jihan Mosalim, who is here, Jihan, say hello to everyone. There it is. Jihan is gonna be teaching a fantastic workshop that is around the subject of GFP and see things that are not there. Jihan, you can speak, please, go ahead.
Yes, just a little detail. Some things might change
Okay.
that's listed on the workshop. So maybe maybe you can visit a bit like in a few days or something. So you have the actual material because it's going to change a little bit. So
perfect. No problem. So then register for that. There's a link. Let me just put the link here. So if you haven't registered yet, I'm gonna put, give me a second. I'm in the other screen right now. Workshops. Register. Here it is, the link. If you want to register, you can go right away. There's in the chat the link. The colloquium is in August 21 at 2 p.m. So it's a 2 hours workshop. We will put the materials in the website, so you will receive those materials. And believe me, they're a lot of fun to do. So come and visit us. Jihan will be there. I will be controlling in the back wherever seeing technical issues, etc., etc. So thank you again so much. Thank you to the museum for work with us, to the guides, to the volunteer guides. You guys are done such an amazing, amazing work. It's such a pleasure to work with all your knowledge and the stuff you brought to the table. I learned so much from each one of you. It has been really, really an honor. Thanks to our speakers, especially today to Melina and all the speakers that participate in this series. We are extremely happy for that. And Stefan, your turn now. Thank you.
I have only similar things to say. I'm so grateful to our presenters, our speakers today, and everyone. If this is your first colloquium or this is your fifth colloquium, thank you for making this a very enjoyable community feeling of public discussion, and you've helped to create something new. Grateful to Convergence for all the work that they have done. Please, if there's one message we keep saying, please go to the Convergence website, visit the tours, take a workshop, enjoy some art, and I'll add my own last link here. Please come and see one of our volunteer guides and enjoy the work that they do. Go see some live art, visit your galleries, visit the museum, it will nourish you. So thank you everyone for being here. It was an absolute honor.
Thank you.
Thank you.
Thank you, everybody. Goodbye.
Bye. Bye-bye.
Evening. Have a great evening. Thanks, everybody.